Okay, this is the Bioengineering 599J spring quarter, the first seminar, and I tried to schedule student presentation once a year minimum. And about 3 weeks ago, we had presentations by 2 postdocs, and today we have 1 presentation by current student and 1 presentation by immediate past student. And toward the end of this quarter, May 23rd, there is another seminar by Dr. Roger Johnson, who was a former student about 7 years ago, 6 years ago, who has been promoted to associate professor with tenure at Marquette. So he can tell you about what is it like to be a professor and trying to get a tenure in an academic setting. So the first presentation today will be by Lisa Vista. She came to University of Washington in September 1988. Sorry, 1998. So it has been about 3.5 years, and she got her BS degree in chemistry in 1998 from Eastern Washington University, and since summer of 1999 she has been working with Professor C.C. Giachelli. And Lisa is going to give us a presentation with the title of Aortic Valve Interstitial Cells in Cardiac Valve Tissue Engineering. Lisa.
Thank you, Dr. Kim. The cardiac valve tissue engineering project is a relatively new thrust in our laboratory, and my contribution to this project has been on the study of the aortic valve interstitial cells, which is the main cellular component in the heart valve. The current therapy for heart valve disease is heart valve replacement. Today, approximately 170,000 valves are replaced each year worldwide, where the mitral and aortic valves are the most commonly diseased valves by such conditions as rheumatic valve disease and endocarditis, 2 diseases which are associated with bacterial infection, age-related degeneration, and dystrophic calcification, which is the abnormal deposit of calcium salts in soft tissues. Such diseases lead to stenosis, or a narrowing of the heart valve orifice, and regurgitation or leaking of the valve. The current heart valve substitutes are either mechanical or tissue bioprosthetic valves. This is an example of a tilting disc mechanical valve. Mechanical valves are made of materials such as pyrolytic carbon, titanium, and other metals. And tissue bioprosthetic valves are made from glutaraldehyde fixed porcine aortic valves or glutaraldehyde fixed bovine pericardium. And while these valves have improved the lives and saved many lives of people, they still have many problems in that they fail at a rate of 50 to 60% within 10 years. The most common mechanism of failure for mechanical valves is thrombosis or blood clotting. The most common mechanism of failure for tissue bioprosthetic valves is calcification, in which the leaves essentially turn into bone, become brittle, and are thus non-functional. So an attractive alternative to heart valve, to these current valve substitutes, is through tissue engineering, where a biodegradable polymer is usually used as a structural support for the cells, which are then introduced into the scaffold and are allowed to form and synthesize their own extracellular matrix, as indicated here in this scanning electron micrograph. And eventually the cells are able to form a complete tissue where the biodegradable scaffold has completely degraded. Tissue engineered valves offer some potential advantages over current valve replacements. One, in that the surface would be endothelialized, which would reduce the chance for thrombogenicity. Secondly, since this is a living valve construct, the cells would have the potential to synthesize their own matrix and remodel and repair it. Next, they would have the appropriate extracellular matrix composition and deposition, which is very important for heart valve function. They would have a potential for growth, which is very important in children who receive heart valve replacements. Among other potential advantages of non-immunological response, it would have stable mechanical properties, more natural hemodynamics. And finally, we could potentially manipulate these valves genetically so that they would have resistance to thrombosis, calcification, and infection. There are a couple of groups who have shown feasibility for heart valve tissue engineering, and one is the Advanced Tissue Sciences, where they constructed this valve here from a decellularized porcine valve scaffold, which they seeded with human dermal fibroblasts. And they reported that the human fibroblasts were mitotic and were able to synthesize their own extracellular matrix proteins. Another group, which is the collaboration between Mayer and Vacanti at Children's Hospital in Boston and Robert Langer at MIT, showed feasibility of tissue engineering valves using a number of different biodegradable valve scaffolds. However, they have reported small sample sizes, no elastin or little elastin is incorporated into their valves, where elastin is very important in native valve function. They offer no statistics, no long-term studies, and their valves are only operate in the pulmonary position when implanted in sheep, and don't function in the aortic position, which has higher pressures. But one interesting outcome of their work was the fact that they determined that cell origin affects the outcome and final function of the tissue engineered valves. Specifically, they found that the arterial wall myofibroblasts performed superiorly to dermal fibroblasts when seeded into the scaffolds. So the idea that the cell origin affects the outcome and performance of tissue engineered valves led us to the following hypotheses. So we believe the valve interstitial cell, or VIC, which is the main cellular component in the native or natural valve, developed from a lineage that is unique from that of fibroblasts or smooth muscle cells, and thus perform functions that would be specific to the maintenance and function of the heart valve. So using the valve interstitial cells as a cell source for a tissue engineered valve could provide us model design principles. This would hopefully yield a gold standard valve construct. And then down the road, we could look for possible stem cells or progenitor populations of the true valve cell that could be used for an autologous cell source. So we isolated the valve interstitial cell from both bovine and baboon aortic valves, where this is a valve that has been dissected longitudinally, and this person's finger is stuck in one of the cusps of the valve. So we dissected out single valve leaflets, where this is the cut edge, and this would be the free edge. If one takes a cross-section of this single valve leaflet, you would see that the valve is composed of three main layers, namely the fibrosa, which is composed mostly of collagen, the spongiosa, which is composed mostly of glycosaminoglycans, and the ventricularis, which is composed of collagen and elastin. And this entire valve is covered with a single layer of endothelial cells. So if we remove the endothelial layer by scraping, and we can do an enzymatic digestion to essentially isolate the valve interstitial cell. So if we look at a histological view of the valve, once again you can see the three layers, the fibrosa, the spongiosa, and the ventricularis, where the blue-green color is the stain for collagen, and the pink is the stain for the cytoplasm of the cells, where you can see the cells are resident throughout all three layers. So this valve interstitial cell is fairly understudied. It is thought to have characteristics both of fibroblasts and smooth muscle cells. So like a fibroblast, it is secretory and has prominent Golgi apparati. When grown to post-confluence, the cells demonstrate an orthogonal overgrowth pattern, and the cells have no basal lamina. However, like a smooth muscle cells, the cells are contractile and have prominent microfilaments and demonstrate gap junction communication. These cells develop from the endocardial cells of the primary heart tube during development through an epithelial mesenchymal transformation. They are thought to be reparative and may also be innervated. In our laboratory, we found that these cells proliferate in response to platelet-derived growth factor, but not basic fibroblast growth factor, which indicates these cells are, again, distinct from fibroblasts. And in addition, a note here that we also found that they can mineralize their matrix in the presence of elevated phosphate, which indicates these cells may play a potential role in valve calcification of native leaflets. So in looking at protein expression and compared these cells to bovine smooth muscle cells, we found a very similar protein expression pattern. We found that the bovine valve interstitial cells express both alpha smooth muscle actin and SM22 alpha. These are two markers that are traditionally indicative of the smooth muscle cells. When we stain for alpha smooth muscle actin in cell culture, it's a little difficult to see here, but we saw that the cells have filamentous actin, which is oriented in the long axis of the cell, which is very similar to what is seen in the smooth muscle cells. So we did a little bit of phenotypic study of the valve interstitial cells, but we also wanted to probe and ask how does the valve interstitial cell interact with its environment? Because we have a hypothesis that if we have a tissue engineering scaffold modified with specific peptides that can communicate with the cell, this would encourage the cell's survival, proliferation, migration, and essentially, finally, tissue formation. So we started to investigate what could we use that would talk specifically to the valve cell to initiate these types of processes, and so that we could rationally design this scaffold. And the first way we went about doing it is this assay we use in the laboratory called the adhesion assay, where we coat different protein ligands at 4 degrees overnight, and then we have a blocking step for non-specific binding. We plate the cells in triplicates and allowed them to adhere for 1 hour at 37 degrees. Then we wash off the non-adherent cells, fix the adherent cells in paraformaldehyde, stain the cells with toluidine blue, and then we solubilize this stain in SDS, and then the solubilized stain is quantitated with a spectrophotometer. So in this experiment, we looked at the adhesion of the valve interstitial cell to a number of different protein extracellular matrix ligands, where increased absorbance readings means increased cell adhesion. So we found that fibronectin, osteopontin, collagen, and laminin all support adhesion of the valve interstitial cells. However, we wanted to investigate further how exactly are these cells interacting with the protein ligands. And that's when we started to ask ourselves, what is the cell's integrin profile? And integrin is a dimeric transmembrane protein receptor composed of an alpha and a beta subunit, where the extracellular domain binds and interacts with the extracellular matrix, and the cytoplasmic domain interacts with the cytoskeleton of the cell and transduces signals to the cell. So in using FACS analysis with fluorescently labeled antibodies to the integrins, where a positive expression is indicated by a shift in the fluorescent peak, we found that the cells expressed integrin alpha V beta 3 along with a number of beta 1 antibodies. And we were particularly interested in this integrin called alpha 9 beta 1, which we further confirmed expression through Western blot. So why do we care about this integrin alpha 9 beta 1? First, this is the first report of its presence in valves, and so we wanted to further elucidate what is the function of this integrin in the valve. Next, the expression is mainly restricted to the epithelium, so we thought that this could be a particular valve cell lineage marker. Next, this integrin mediates adhesion, which is it's non-RGD mediated. And this is particularly important in cardiovascular tissue engineering, where the tissue engineered constructs would be in direct contact with the circulation. This is because platelets express alpha V beta 3, which do recognize this RGD motif, so that we could promote valve interstitial cell adhesion to tissue engineering scaffolds by utilizing this integrin without promoting thrombogenesis or platelet attachment. So, and in addition, we wanted to ensure that the expression that we found of alpha 9 beta 1 was not simply an artifact of cell culture or an artifact or specific to the species bovine. So we looked in baboons, where we were able to, using reverse transcription polymerase chain reaction, we were able to amplify an alpha 9 subunit cDNA both in baboon cell culture and in whole tissue. And this work was confirmed by sequencing. So this integrin, like I said, is expressed in the squamous and airway epithelium, some smooth and skeletal muscle, hepatocytes, and neutrophils. It has 3 known ligands, tenascin C, VCAM1, and osteopontin. And a mouse knockout develops congenital chylothorax and has respiratory failure and dies within 6 to 12 days. So what are the potential functions of this integrin in the body? It is thought that it has an oncofetal function and that it is seen expressed during fetal development and also in cancerous tissues, although it is correlated with development of a more mature tissue phenotype rather than very early organ. The binding actually being mediated through the alpha-9 beta-1 antibody or integrin. So we did blocking experiments to answer that question, and we pre-incubated the cells either with this peptide ligand or the alpha-9 beta-1 antibody, such that if the integrins are all bound up by the antibody or the peptide, they would not be available to bind to the osteopontin ligand. So in this experiment, the cells were pre-incubated either with no antibody, an antibody to alpha-9 beta-1, or in blue, an antibody to beta-1 integrins. So, and then we plated the cells on either 30N osteopontin or the 2 mutants or a PBS control, and we see that in the presence, in green, in the presence of the alpha-9 beta-1 antibody or the beta-1 antibodies, on both of them means that the adhesion was inhibited. So we were able to determine that the adhesion to the N-terminus is actually mediated by alpha-9 beta-1 integrin. But then we wanted to see, is the integrin actually interacting with this SVVY-GLR motif? So we pre-incubated the cells either with no peptide or increasing concentrations of either the peptide of interest or a scrambled negative control peptide, and we saw a nice dose-responsive inhibition of cell adhesion. So I'll back up just a second and kind of conclude on those two slides. Is that so we were able to determine that this peptide could be used on a tissue engineering scaffold, whether it would be immobilized on a tissue engineering scaffold, to support adhesion of these cells through interaction with alpha-9 beta-1. However, we wanted to also determine if the alpha-9 beta-1 integrin could mediate migration in these cells, which is also important for tissue formation. And so what we did in the migration assay was to coat the bottom of these filters with a protein, and these filters have 8-micron pores. And we coat the protein for 1 hour at 37 degrees. And then we fill the bottom of the well with media, and then we add the cells to the top of the insert, and the cells are allowed to migrate for 24 hours. And then at 24 hours, the cells are labeled with this Cyto11 nuclear stain, which allows us to visualize the cells using an inverted fluorescent microscope, where we count 3 random fields per high-powered field, or 3 random high-powered fields per membrane. So in this adhesion assay, we pre-incubated cells either with no antibody, a beta-1 antibody, or an alpha-V beta-3 antibody, and plated the cells either on 30N osteopontin, 30N RGE, or PBS or GST controls. And so when we look at the 30N osteopontin, or migration to the 30N osteopontin, you see in the presence of no antibody, the cells were able to migrate to the 30N fragment. However, in the presence of the beta-1 antibody, they were still able to migrate, whereas the alpha-V beta-3 antibody completely knocked out migration, which indicates that, well, in addition, they were able to migrate to the 30N mutated fragment, which indicates that the integrin alpha-9 beta-1 does not mediate the migration of these cells to osteopontin. And in this last experiment, we labeled the cells for proliferation using a tritiated thymidine uptake, which is a radiolabeled nucleotide that is incorporated into the cells during proliferation. And so we plated the cells either on poly-D-lysine, which is a negative control, which does not interact with the cells through integrins, the positive control tenascin C, the 30N fragment again, and the mutated form. And so here we were able to simulate proliferation on the 30N that still maintains its RGD, but on the mutated form we were not able to simulate proliferation. So this further indicates that the integrin alpha-9 beta-1 is actually not capable of stimulating proliferation on osteopontin. So in summary, we have isolated and characterized the adult valve interstitial cell phenotype through protein expression, the adhesive ligand's integrin profile, and its growth response to serum and growth factors. We found that the valve interstitial cell express alpha-9 beta-1 both in culture and in the native tissue, and that they mediate adhesion to SVVY-GLR through the alpha-9 beta-1 integrin. However, this integrin does not mediate migration or proliferation on the 30N RGE osteopontin. So as far as manipulating and using this peptide interaction with the alpha-9 beta-1 integrin, we could perhaps enhance adhesion to a non-adhesive tissue engineering scaffold, but it doesn't look like we could depend on this interaction for inducing proliferation or migration. So the future plans are to select a scaffold and immobilize this peptide and see if we can increase adhesion. And secondly, we'd like to look at microarray analysis such that we can look at further look for lineage markers of the valve cells and compared with fibroblasts and smooth muscle cells. And finally, I've begun some work on genetic engineering these cells to express an anti-calcific protein, which could also be potentially advantageous in tissue engineering. I'd like to thank Kip Hauk in the UWeb microscopy lab, the UW immunology fax facility, Bart at the slaughterhouse, he provides our bovine samples, UWeb and the National Science Foundation and the GAN Fellowship for funding, and of course the Jackelli Lab Group for their support.
Okay, thank you. I'd like to ask you withhold your questions until the second presentation is done. Okay. So, next presenter is Dr. David Nutley. He got his PhD or defended his PhD about 3 weeks ago in March, and in bioengineering. So he received his BS degree in bioengineering from Syracuse, and he came to UW after that. And he finished PhD in bioengineering in March 2002. Currently, he is a research associate in the mechanical engineering department at UW. And his research at the Applied Biomechanics Lab is focused on understanding the mechanics of the developing sign at the system and cellular levels. And David, I think you served as a president of BESA in 1996, 1997. So you know how long he has been around. He came here in 1995. So, David.
Thank you very much. I have been here quite a while, and so I'm not going to share with you everything I've learned here at the UW. What I'm going to share this afternoon is a subset of my dissertation research, looking at taking the experimental data that we collected and trying to put that into a useful model whereby we can positively affect child injury prevention. The research that I performed on the maturing spine used a number of different models, animal models and human models, to try and assess how development affects the mechanics of the spine. The reason that we were interested in that is approximately 10,000 children each year incur some sort of spinal cord injury. 2,000 of those are fatal, and the rest are left with debilitating paralysis or resulting injury. And so we really wanted to try and get at those problems in children which are affected by everything from automotive to sport and fall incidents. I'd like to start out by thanking my co-authors on this project. One co-author who didn't get up here, but actually I should be up here, I need to mention Dr. William Ledoux did a lot of the mesh generation for the model that I'm going to be presenting. So quick impetus for our investigation. Why are we looking at pediatric injuries, or why are we looking at how the spine develops? Well, a brief look into the anatomy. This is a cross-section of the spine that you see here, of a 2-year-old spine. And if this were an adult spine, it would be a complete ring of bone. However, in the 2-year-old, there are synchondroses here, which are cartilaginous in nature, and allow movement between these relative bones of the 2-year-old vertebral body. Those don't fuse until about 7 years of age, and at 7 years of age, growth plates, which are as well cartilaginous, appear on the superior and inferior aspects of each vertebral body, and they produce the longitudinal or height growth that is commonly experienced in adolescence. And so as you can see, these different anatomical and morphological changes in the tissues that make up the spine are going to have a very different effect on the mechanical response of those tissues. And we see that when we look at the epidemiology literature as well. The predominance of injuries to children are very unique. In adults, we see a lot of lower cervical spine injuries, so lower neck injuries. In children, though, very young children here, less than 8 years of age, we see upper cervical spine injuries. And so that was really interesting to us. What happens in the upper cervical spine maturation that makes that sort of the weakest link in the chain. And so there's an injury predominance and injury patterns in children that make us stop and think, maybe there's some link between the anatomy that we're not understanding, and that is the functional and the failure mechanics. So how do the mechanics of these tissues behave? And if we can understand that and try and model that, then we can get at a number of different clinical outcomes. We can clinically assess injuries a little bit better, and also understand basic science. How, as cartilage becomes bone, what are the mechanical changes that come about in those tissues? As well, and primarily for this lecture, we can understand injury prevention strategies. If we know how an injury occurs, we can come about and look at a strategy that can minimize that injury in children. So, reverting all the way back to early engineering, what do? When we want to try and understand a system, we model it. We try and use some sort of physical or computational model to understand how that system behaves. For all of our experimental work, we used baboons, likely those that the cadaver version of the ones that Lisa spoke about. And we did cadaveric tests on the baboon cervical spine, and we performed a number of experimental studies to try and understand how, as those tissues develop, the mechanical characteristics change. And then we apply those characteristics to both computational models as well as anthropomorphic test dummies, which are all familiar probably with in automobile situations, to try and give them real values so that they can assess something when they receive a crash input. And so it's this relationship that I'm going to be talking about in terms of a computational model development. And really, what I'd like to walk you through is the process of developing this model, the choices that we made in terms of the type of model, and those aspects, how they were affected by the experimental data that we collected. So in mechanics modeling, there are a number of different models that you could choose, statistical or empirical, finite element, or rigid body models. From our experimental data, we saw that developing vertebral bone is 10 times stiffer than the soft tissues between. So since there was that 10 times disparity, we felt that we would be able to take a very first order approximation of this system as a rigid body, where we assume that the vertebrae, which you can see here, all as different colors here, are rigid bodies, modeled as shell elements, and then we model the mechanics between those shell elements as a set of lumped parameters. So once we chose a model, then we chose a model strategy, we need to now define the geometry, define the mechanics between with experimental data, and then turn around and say, does this model make sense? Is it validated? Slicing this computational model a different way to give you another feel for the aspects that are involved in modeling the spine. There are two main criteria: the functional mechanics, which is sort of the everyday functional mechanical response of the spine as you're walking around, and then there's the failure mechanics. When you take the spine to excessive levels of force or displacement, how do those tissues behave? And as you can all sort of move your head around, you can understand that there are compressive, tensile, bending, and shear elements that all need to be incorporated into this model to really get the mechanics of the head and neck motion appropriate for the spine. As well as changing all of these parameters, we're also changing with age. So we're looking at how these different parameters develop with age, and they may not be all be the same. Concerning the failure mechanics, or the tolerance criteria, I put some charts over on the right-hand side here, and these charts represent the current tolerance criteria for crash test dummy neck loads that were used as recent as 2 years ago. 2 years ago, we had just a very little data in terms of what we called failure in these crash test dummies. So we put these dummies into cars, crashed the cars, and we used one number for all of these criteria, for each aged individual. And I'd like to show you throughout this talk that we've gotten a lot more sophisticated since then. So first, building the model. So we're going to start out with geometry. Our goal is to eventually build a whole host of models, 3, 6, 9, 12-year-old models. But as you saw in the first slide, that the geometry changes. What is bone and what is cartilage changes throughout maturation. We decided to take our first approach looking at a 6-year-old model. So we took a 6-year-old male human, we scanned that individual, and then we created polyline slices. So we created a 3-dimensional geometry of that entire CT scan. We then created an axisymmetric mesh, trying to minimize the number of nodes so we could increase our run times and really do a lot of parametric analyses. And I'll show you sort of how we created those meshes. In yellow, you can see from the previous slide those polylines, so you can see the lines here. And we developed the mesh around those lines, so we incorporated all the geometry of those vertebrae, and trying to do that in as few nodes as possible. We then took the vertebrae here, and I show C3 and C4 here, and we stack them up into an idealized model here. This is looking anteriorly. You're looking at the vertebral body anteriorly here. A lateral view, you see the spinous process sticking out back, which is what you can feel on the back of your neck if you feel back, and anteriorly the vertebral bodies. And what you see from this idealized model, and you could also see on the CTs, is that there are many different interaction points between these vertebrae which affect the mechanical performance. There are facet joints here. There's an anterior connection between the vertebrae of the uncovertebral joints. As well, there's soft tissues and an intervertebral disc, which further affect the mechanics. So we need to come up with a way to define that mechanics in all the directions that we discussed. And what I'm going to present today is just one direction of that model, so that we can sort of all get a good feel for how the model was developed. To model those intervertebral mechanics, we chose lumped parameter components. And we really needed to figure out a way, how are we going to model those? There's a bunch of different ways you can model them. It's just as linear elements, elastic, no rate dependence. But we decided to turn back to our experimental data. And what we saw is on the load displacement curve, this is load here, displacement here. And as you increase in displacement, you see a nonlinear, and it almost appears exponential, elastic response of these tissues. So that led us down the road of selecting a nonlinear elastic model. And then we wanted to know, is it important in these tissues to include a viscous behavior? Is there a time-dependent behavior to the tissues? Well, in fact, when you run a step input and look at the stress relaxation of the tissues in orange here, the force relaxes over time. So there is that viscous or time-dependent behavior with rate. And so we needed to come up with discrete elements that were nonlinear viscoelastic. The simplest approach to that is to define those as a Kelvin-Voigt solid. Kelvin-Voigt solid here is a spring and a dashpot in parallel with one another. Better than all the models before it, we've decided to model each of those components as nonlinear. So we will apply a nonlinear rate dependence and a nonlinear elastic response to the tissues. We decided to represent these elements and get all the data from curve fitting our experimental data. And the curve fits you'll see here are an exponential of the stress-strain relationship. And then for the dashpot coefficients, we calculated the hysteresis energy from a stress relaxation loading-unloading profile of constant rate. And from that hysteresis energy, we can calculate the exact coefficient for damping at a specific rate. Now I'm going to show a little bit of the experimental data to just sort of give you a feel for where this model will go in the future. We're specifically looking at a 6-year-old model right now. However, vertebral stiffness, so this is a vertebral body stiffness. In the upper right-hand corner, you'll see the little icon of a vertebrae here with compression. So vertebral stiffness in compression significantly increases with skeletal maturation. So as you get older to adulthood, there's a significant increase. Not terribly unexpected. And I point out the 6-year-old age range to show you that there were only a few specimens there, and so our modeling effort actually involves the use of our line through the middle here to try and best approximate those 6-year-old properties instead of using one specimen or two specimens depending on what we might have at that age range. We did a subsequent study where we looked at the mechanics of the tissues between two vertebrae. So now I'm compressing on two vertebrae and all the tissues between them. And what you'll see is, again, the similar shape curve here where the stiffness in compression increases with maturation. And again, I point out that the 6-year-old, we did only have one specimen in this group. And so it would be very difficult to build a model around one specimen. And so we thought it best to model the entire relationship. And finally, I show tensile data here. So now we're pulling on that same specimen that we pulled on in the previous test. And the tensile stiffness significantly increases with developmental age. Again, I'll show the 6-year-old here. And this is what we're going to be primarily talking about the rest of the talk, the mechanics of this 6-year-old in tension. So taking the lump parameter approach, I curve fit each of the data sets. And you can see the red data here is the experimental data of two specimens, and the black line is the fit between those two specimens. So I think that the exponential fit very closely approximates our experimental data for the 6-year-old. For the adult, where we have a longer toe region and a steeper stiffness, might miss a little bit down here, but I still think that we have a really good approximation of the exponential fit to our experimental data. When calculating the hysteresis energy and the dashpot coefficients, we see really nice increases in the dashpot coefficient with maturation. As well, a decrease with loading rate, and that was all expected. So now I'm going to sort of jump to a validation of this model. What I did is I took the tensile stress-strain relationships from all the specimens that we had, and I came up with adult, 12-year-old, 6-year-old, and 3-year-old mean curves. So I had the mean and standard deviation of each of those experimental tests that we ran, and you can see those presented right here. I'm going to show the next slide here. We'll remove the mean data. So this is real experimental data that we had. I'm removing the experimental data, and now in the next slide here, I'm going to show our model fit, exponential fits that came about through the model. And this is for tension only, but we've done this all the other directions as well. And you'll see that within one standard deviation, our model predicts the stress-strain response extremely well. Now back to the endpoints. So those curves continued going and going, but a model really needs endpoints. We need to have some value where someone who's looking at an anthropomorphic test dummy or a computational model can say a child would have been injured at this point. And so I think one of the strongest results of my experimental work was to come up with criteria for ending that modeling effort. These are the previous values for tension and compression that I showed you on an earlier slide. And in green are the values that we came up with from our experimental work for the endpoints in force across age. And the FMVSS is the Federal Motor Vehicle Safety Standard 208, which came out last year. And you can see that the numbers are very close to the numbers that we came up with in our study. And in fact, that's not by mistake, because our data was used in deriving those criteria. And so I'm extremely proud of the fact that the new Federal Motor Vehicle Safety Standard includes data that I was able to collect. And so hopefully those will be criteria that will enhance injury prevention from dummies. So in summary, we've used nonlinear spring and elastic responses to model the mechanics between two vertebrae. Nonlinear dashpots, which were able to be rate dependent. That's a new thing to computational modeling. And also we've developed criteria whereby the pediatric spine can be assessed in anthropomorphic test dummies as well as computational models to assess when a child may have been injured. And just in brief conclusion here, the modeling of the maturing spine, I believe it's essential to have a nonlinear viscoelastic response to those tissues. It's not an easy model, but it makes for the most complete and accurate model. And that the model that we've created is able to be tuned by age, loading rate, as well as other factors. And I really look forward to, in the very near future, using this model to facilitate injury prevention for children. I'd like to thank the National Highway Traffic Safety Administration for their funding, as well as Centers for Disease Control and Prevention, and the UW Primate Center for the tissues that we tested. Thank you very much for your attention.
Now, questions to either David or Lisa? Yes.
I have actually two questions. One is, did you take into account neck muscles?
No, these are all of our specimens are cadaver specimens, and so they have passive musculature. Our next generation model has sternocleidomastoid, and it has a few muscle connections, and so it will have muscle tone in the next generation, but we don't currently have that now.
So it means that you are underestimating the actual value of that for...
Yes, probably. But what we believe is we're getting the worst case scenario. In a lot of automotive accidents, the time to peak load is less than muscle contraction times, some on the order of 10 milliseconds. So they might not even have full contraction anyway. So we're modeling the worst case scenario here.
And was there any difference? I mean, have you looked at males versus females? I know we looked at different ages, but how was the gender difference?
Yeah, we did a subsequent study that looked at male and female differences in vertebral bone, and we actually found that when you normalize by size, females in maturation are stronger than males. Their curve ramps up a little bit steeper than the male curve, whereas the male curve is a little bit more shallow. And we believe that that may be one of the indicators of early onset osteoporosis, that they ramp up much faster in their mechanical properties and the orientation of their trabeculae, and that it leads to the early fall off. I mean, this is just, we're hypothesizing on that. But yeah, there are differences between males and females, clearly.
Other questions? What was the difficulty in doing the experimental and computational work and trying to make sense out of both?
Really, the most difficult thing was setting up your experiments initially so that you collected the correct data for the computational models. I didn't show this, but we ran a whole battery of experimental tests where we did cyclic loading, triangle pulse loading, so we got constant loading rates, loading on, loading off, various loading rate tests, stress relaxation tests, and so we really ran a whole battery of tests. And I think one of the limitations was we collected too much experimental data for our own good, but it's been nice to be able to sort of look back at that data for validation purposes. But really, my advice would be, if you're going to end up using experimental data to model, make sure you ask the question before you start collecting your experimental data so that you know you're collecting the right data.
Any other questions? Or questions to Lisa? When are you finishing?
Hopefully June 2003.
June 2003. That's within 5 years. Okay. Okay. Question there? Repeat the question.
The question was, have we considered using human valve interstitial cells? Is that right? We would love to, but it's just so difficult to obtain those samples. And usually, if you can't obtain human valve interstitial cell samples, it's because the valves were diseased in the first place. And so that's why we're still using the models of bovine and baboon, which is close to human, but not quite.
So you had to go through all the animal care committees approval?
Yes.
Okay. Which is not straightforward.
Not easy, no.
It's a lot of time and care. Any last question? Okay. Thank you, Lisa.